Okay, so now we have finished our dilutions for our challenging samples. We've picked out one high quantity sample and one low quantity sample. So now we are on page 76 of the lab manual. So we've diluted our samples down to our either 0.8 nanograms, or we have our low quantity sample where we were going to be inputting less than 50 picograms. So I have our two samples here. We've already diluted them, so they're ready to go. So the next thing we need to do is make up our PCR master mix. So first thing we typically do is we decide how many reactions do we need to make. Well, we have two samples. We need to include a positive control, and we need to include a negative control. So typically what I like to do is I count up how many reactions I need, so 4, and I go ahead and add one extra reaction just to make sure. So basically what we're gonna do is multiply all of these volumes by 5. So we have 7.5 times 5, which equals 30 microliters, and 2.5 times 5, oops, does not equal 30, equals 37.5, and 2.5 times 5 equals 12.5. So that's going to be our master mix. So if we take a look at our box called Globofiler PCR reagents, this is going to include 3 tubes in the pre-PCR box. So this yellow tube is our control DNA. This is control DNA 007, so we'll use this for our positive control. We have Globofiler master mix in the blue tube, which contains things like the Taq polymerase, the buffers, the DNTPs, magnesium, all of those good things. And then most importantly, in this kind of teal turquoise tube, we have the primer mix. So hopefully you can see on the video, but this is kind of a pink color. So the primer mix typically has a color to it, whereas the master mix itself doesn't. Okay, so now that we know how we need to make up our master mix, I'm going to get a blank tube, has nothing in it, and I'm going to add the primer mix and master mix components. Okay, so 37.5, grab another pipette. These have been previously vortexed and spun down, both the master mix and the primer mix. Okay, master mix in, now 12.5 of primer mix. Again, it's really important to vortex, especially the primer mix, prior to making your master mix, because we want to make sure it's a homogeneous mixture. Alright, so typically you'll label this tube master mix. Let's go ahead and vortex this up to make sure that, again, we have a homogeneous mix of our master mix components and our primer mix. We would typically spin this down really quickly. Okay, good to go. So the next step that we would do is find our PCR strip tube. It's got these domed cap lids. Typically what I like to do is, so we have eight wells here. We only have 4 samples, but I know that we're gonna need to include an allelic ladder to run with these samples. So I typically leave this first well blank, and I start with sample 1 and well 2, sample 2 and well 3, and then my positive and then my negative. So our total reaction volume is going to be 25. 10 microliters of that is going to be our mixture of our master mix and primer mix. So we can go ahead and pipette our master mix into these wells. So I will adjust my pipette to 10. And pipette that in. There's nothing in here right now, so I don't need to change my pipette tips. Okay, good to go. Now, I've already diluted these down to where I can put in 15 microliters of this dilution, and it will equal out to 0.8 nanograms for sample 1, which is our high quantity sample. And for sample 2, if I put in all 15 nanograms, it will be less than, if I put in all 15 microliters, it will be less than 50 picograms. So this is our low quantity sample. So we will go ahead and put 15 in. This is where you definitely need to be changing your pipette tips. So sample 1 goes in well 2. Sample 2 is gonna go in well 3. Okay, positive control. This is where we have to change things up a little bit. Positive control, it's been previously vortexed and spun down. This positive control is at a concentration of 0.1 nanograms per microliter. So to get to 0.8 nanograms, we need to put in 8 microliters of this, and then add in an additional 7 microliters of water just to cue our volume up to 25. So let's do that. Eight microliters of positive control into well 4, and then 7 microliters of water into well 4. So negative control, hopefully you've guessed it, but we are just going to be putting water into this well. We still need it to be at the 25 microliters, but we don't want to include any DNA. We want to make sure that all of our PCR reagents are clean and free of any exogenous DNA. Okay, 15 microliters of water in. We are good to go. We sealed this bad boy up. I usually label on the tube like a 1 or something to kind of denote where my samples started. Typically we would have a sample sheet as well. Now we're going to take this to the PCR thermocycler. Okay, so now we have our samples that we've added the PCR master mix our diluted samples. We have our positive and our negative. So now we're gonna put this on the thermocycler to initiate the PCR itself. So we're gonna use the ProFlex today. So basically all we would do is, since we're not using a plate, we would use this guy and we would put this strip in, shut it, lock it, tell it to set up a run, open a method. Hopefully it's already been saved. We're running Globofiler, so that is what I searched for and found. If you want to confirm in your lab notebook on page 99, you can double check and make sure that the cycling parameters are correct. We would hit verify block. Yes, we only have one block. Some instruments can have multiple blocks that you can control multiple at one time. And we press start run. And then it's going to give us a kind of estimated time remaining, which for this says 1 hour and 20 minutes. Okay, so now our PCR products have been amplified. So we're going to take them off thermocycler, and we are going to bring them over to set them up for the CE. So here is our CE plate. If you take a look at page 100 in your lab manual, you will see the way that we need to prepare our Globofiler PCR samples prior to capillary electrophoresis. So in each sample, we need to include high dye, which we get from the freezer, and then size standard. So we are going to make a mixture of high dye plus Liz 600 size standard. So remember, we're adding the allelic ladder. So we used to only have 4 samples, now we have 5. So again, I'm going to account for overages and calculate for 6 total samples. So let me make the master mix. So I need 57.6 microliters of high dye. And I need 2.4 microliters of Liz 600. So I'm going to vortex this up, and then I'm going to spin down. Okay, 2.4. You want to make sure that you have pipetted up some of this size standard, and you'll be able to visualize it being pipetted into your high dye mixture as well. I'm going to go ahead and label this master mix. Then we are going to take the septa off of our CE plate. Okay, so now we are going to vortex up our master mix, spin it down, and then we are going to pipette 10 microliters of it into our wells where we are going to put in our PCR products. Again, we don't need to change pipettes here because there's nothing in these wells. So because all 8 capillaries are going to be injected, we want to make sure we have liquid in all of the wells, even though we're not running samples in all of them. So we usually just put in high dye. Next thing we need to do is we need to pipette in our PCR samples. So again, we're going to vortex, flick down, remove this cap. We can use a multi-channel pipette. We're gonna set it to 1 microliter. Remember to leave sample, or to leave well 1 blank. That's where we're going to put the allelic ladder. So I'm going to grab our 4 samples and then pipette in. And then I'm going to find a box that comes with Globofiler. It's called the allelic ladder. So we want to make sure we protect the allelic ladder from light, so that's why it's in this film canister looking thing. But again, we're going to vortex, we're going to spin down, and then we are going to treat our allelic ladder just how we treated our other samples, and we are going to pipette in 1 microliter of it. It's very much just like PCR products, except instead of containing a maximum of 2 alleles per loci, it contains all of the possible alleles for that particular locus. So you want to make sure that you get this pipetted in. You need at least 1 allelic ladder per 3 injections. So an injection is just 1 column. Now I'm going to go ahead and mark that we've used this column. Then I'm going to put the septa back on. We can vortex briefly, and then we are going to want to spin this down. You want to remove as many bubbles as you can. It's okay if there is still some, because during the denaturation, most of these bubbles are going to dissipate just with that heat. So we take our plate back over to our thermocycler. I'm going to remove this because I don't need it. I don't want this to close all of the way, so I leave it open because I don't want that to burn the septa. Open method. I find the denaturation and snap cool method. Here it is, 3 minutes at 95 degrees, and then infinity at 4 degrees Celsius. This would be considered our snap cool. Same thing, verify the block, start run. So we'll wait for this to finish, and then we will get it onto the CE. Okay, so we have denatured our sample and we let it sit on here for an additional minute to snap cool. So remember, everything that we're doing here is to ensure that our DNA is single-stranded. The high diformamide is going to ensure that. The heating is going to ensure that. And this snap cooling is kind of doing what it sounds like. It's kind of snap cooling everything in place. So we are triply ensuring that our PCR products are single-stranded. So we're done with the thermocycler. Now we need to move on to the CE. I know some of you got to have a tour of the CE before everything went online, but for those of you who didn't, I'm gonna try and do my best to capture it today. Okay, so here is our ABI 3500 genetic analyzer. So I'm gonna press the tray button. This is going to bring everything forward and allow me to load samples. Okay, open this guy up. All right, so right here is the cathode buffer. Remember, this is the positive buffer. This is nearest to where our samples were gonna go, which is here. And over here is the anode buffer. So basically, this right here is the capillary. It's sitting behind this oven door. Everything is going to maintain that 60 degrees Celsius temperature to ensure things are migrating consistently, everything is staying single-stranded, etc., etc. But we have 8 capillaries here, and they are gonna go and inject into these sample columns essentially. So I'm going to be telling the instrument, hey, my samples are in column 8. Shut this. Oh, another note, behind here is where the detection window is. So this is where the laser is shooting onto the kind of open capillary, and it's detecting the fluorescence and recording it. The capillaries then kind of end right here, and they basically just, the end products are going to dump into the anode buffer there. Right here is the polymer, and this is the pump block. So basically, you will hear a kind of a pumping action before every injection. This is filling up those capillaries with brand new polymer, so it doesn't contain any DNA fragments, anything like that. It's fresh new polymer. Okay, so what would we do next? We would take off this, this cassette, and we are going to put in our samples. So again, I'm taking this out and putting the new one in, noting where my samples themselves are. Again, column 8. And then I will replace this. There's a little latch. It can only go in one way. That's very nice. And then I will shut the oven door, or I will shut the entire door. And it will go back and it'll do its little jig. So now let's go to setting everything up on the computer. Hopefully some of this is pretty clear, but basically on the computer we are able to tell how much of everything we have left. Do we have polymer left? Is the anode and cathode buffers, are they still good? How many injections have been done on the capillary array? Etc., etc. So now what I need to do is I need to basically create an injection file. So usually what we do is we edit an existing plate, because we've obviously run plenty of Globofiler plates before. So I would go in and I would find the appropriate column, it's column 8. I'm gonna label what everything is. So the first well, if you remember, is the allelic ladder. Well 2 is sample 1. Well 3 is sample 2. Well 4 is positive control, negative control for well 5. I'm going to get rid of anything that was run before, merely just delete them. And then for these 5 samples, I tell it which assay it's running. So for us, we're running Globofiler assay. This tells it what injection parameters it needs to follow, so how long it needs to inject for, the voltage, things like that. We're going to tell it kind of some file name conventions. And last but not least, we are going to tell it what results group we want it to go into. Another thing that you can do is you can tell it what type of sample everything is. So I could tell it that the sample named allelic ladder is indeed an allelic ladder. And then I could do the same thing for the positive and negative controls. This is going to help us on the back end. Then I'm going to press link plate for run. It's going to go through and it's going to make sure everything is up to date. It's going to make sure that you have the appropriate kind of calibrations in order to run Globofiler. So do you have matrix standards for these dyes, etc., etc. It's going to let you know, hey, I have everything linked on plate A. Nothing is linked on plate B, which is what we want. I usually tell it, can you create me an injection list, just so I can double check. And again, we're confirming that it is going to be running everything in column 8, which it is. And then we would press start run and it would do its thing. Okay, so we have ran our samples on the CE. They have been separated and detected. The CE itself has made a .hid file, so we need to import it into our automatic genotyping software, GeneMapper IDX. So to do this, we are going to hit that edit button, and then we will have a chance to add samples to our list. So you're going to be able to query the samples that you want to add and then add the samples to your list, and then they will show back up here. So now you're going to select all of your analysis parameters. So you're going to select the size standard, which is the Liz 600. You're going to select the panel, which is Globofiler, and you're going to select your analysis method. You will notice that samples are labeled samples, and the allelic ladder is labeled the allelic ladder, the positive control is labeled the positive, and the negative is labeled the negative. When we go into our actual samples, we can actually edit the electrophoregram. I've just briefly shown you how you can right-click on a peak that you know is stutter, and you can delete that. Now I'm showing you what the allelic ladder looks like in GeneMapper IDX. You can see for the blue dye channel that we have all of the loci with the corresponding alleles in those gray bins, and it is the same for the green, yellow, red, and purple loci. Now we are looking at the orange dye channel. If you remember, this is where our size standard is going to be at in all of our samples, including the allelic ladder. This is the size standard Liz 600 that contains known base pair size peaks. Looking at the size match editor, we can confirm the quality of our sizing. You'll take a look here and we know our sizing quality is a 1.0. The peaks themselves are actually converted to a sizing curve through a local southern method. You can see that the R-squared value is very, very close to 1.0. Here I want to show you what the Globofiler panel looks like. So this is from the manufacturer. You will see on the left-hand side that we have the loci or the marker name. We have the dye color, the approximate size range, control alleles. So these are the alleles to be expected from the positive control, the repeat type, whether it's a Y marker or not, and then the allelic ladder alleles that we can expect with each of these markers. Here's a closer look at one of the loci. You will see here that we have bins denoting where our allelic ladder alleles are going to be at for this particular marker. Just to note, those gray bins are the alleles that are expected in the allelic ladder, while the pink reddish bins are potential other alleles that could occur in your sample. Our panel also has stutter thresholds for each of the loci. Here you will see the approximate percentage of minus stutter expected with one particular locus. Here's what the analysis method editor looks like. Again, this is where you're telling it, hey, these are the bins that I want you to look at. Here's the approximate stutter if it's not listed. So here we are looking at, in the software, we can tell it which peaks we want it to call. So we're able to tell it you can call peaks from base pair size ranges of 59 up to 460. So it won't be looking any further than that. This is where we tell the software our analytical and stochastic threshold. The analytical threshold is that heterozygous minimum peak, while the stochastic is that homozygous minimum peak, both up towards the top of the page. These are some other parameters that we can set in our analysis method. An important one I want to point out is the peak height ratio there on the right. This is basically the minimum peak height ratio for the software to call that two peaks do belong to each other.